You're right. I did not put the last button for the introduction, but for everyone, the presentation I just showed, I actually uploaded on YouTube before. So you can still watch it there again if you want. But we are now starting with Roman's talk. And so the most important thing will be on YouTube.
Good. Thanks a lot. All right. So I think now you should see my screen. And just make it so that I can see you guys. Okay. So thank you, Carsten, for the introduction. And I would like to thank the organizers for this seminar series. I think this first session has been a great success, and I've learned a lot from what I've seen, and it's very nice of you to take the time to initiate and organize these seminars. And you've done a terrific job, and I think we are all grateful for that. So I will start my presentation today very similarly to Leticia's last week by telling you that one central question in biology is to understand the emergence and the organization of complex biological systems that rely on the cooperation of specialized entities that have different characteristics and fulfill different functions. And these complex biological systems originated from major transitions in evolution, and they occurred at multiple phenotypic scales. You can find such cooperation and specialization among organelles within a cell, among cell types within an organism, and among individuals within a society. And despite occurring at very different scales, there are commonalities to the major transitions in evolution. And an important one is that they are transition from a pluripotent ancestor that would alternate between different states, and then those different states would be expressed in different specialized elements that would cooperate to function at a new higher phenotypic level. And this is what happened during, for example, the evolution of multicellularity in animals. The ancestral state was a pluripotent unicellular organism that would alternate between different phases of a complex life cycle. And then colonies were formed, and some cells started to stay in one phase of the life cycle. And this gave rise to specialized cell types that would cooperate as basically the first animal. And the emergence of specialization also occurred during the evolution of social life. We find extreme examples in the eusocial insects, the bees, the termites, the ants, and the wasp. And there you have clear specialization because you have division of labor between individuals that are morphologically specialized to perform different functions. And in eusocial Hymenoptera, these are the queen and the worker caste, and they have evolved repeatedly, independently in bees, wasps, and ants. And you have roughly 15,000 species of ants, and they showed great diversity in many aspects, in morphology, colony size, individual size as well, in color and so on, but also in social complexity. You can have very simple ant societies that are composed of a few individuals that are very similar, morphologically speaking, up to very advanced and specialized societies where you have very strong morphological differences between the castes. But all this diversity emerged from a single origin of eusociality. So all ants evolved from a single origin of eusociality. And the subsocial ancestor of ants was some kind of a wasp, a subsocial insect, meaning that's an insect that would do maternal care. And this ancestor would alternate between reproductive and brood care phases. So you can imagine some kind of a wasp that would lay an egg, and then that would be the reproductive phase. And then this egg would hatch into a larva, and then the mother would not lay another egg. but would care for that larva, forage for food, feed that larva. And only once development of the larva would have been completed, then the mother would lay another egg. So this subsocial ancestor was pluripotent. It was able to express both reproduction and brood care. And it's commonly accepted that the two phases of the subsocial cycle have been decoupled over evolutionary times to be expressed at the same time, but in different individuals. And this gave rise to the queen and worker caste that we see in ants. And I'm interested in this transition from pluripotency to specialization. And I studied the mechanisms and the factors that controlled or were involved in that transition, and therefore that were involved in the evolution of female caste, of division of labor, and more generally of eusociality in ants. And this is not a new question. This has attracted a lot of attention for a lot of time. And traditionally, the way to address that question has been to compare the queen and the worker caste. And it has brought tremendous amount of very important information on the evolution of the organization of ant societies and also of eusocial insects. It allowed to identify queen pheromones, for example, that are produced by the queen to communicate their fertility to the workers, and to which the workers respond by inhibiting their own reproduction. It also allowed to identify genes and hormones that control the alternative development of a female larva into either a queen or a worker. But I think it's time to complement that approach with other approaches to study the evolution of the female caste. And I'm saying that because I think there are two main limitations to the comparison of queens and workers. The first one is that the castes are fixed at the adult stage in eusocial Hymenoptera. And that means that you are bound to correlative studies. You cannot manipulate the system. Once a queen is a queen, it's too late. So this goes in the way of doing experimental manipulations to prove causal links, to functionally show the role of one particular mechanism in controlling division of labor, for example. And the second limitation is that the queen and the worker caste differ in many interrelated characteristics. Queens and workers, sure, they differ in reproduction and brood care, but they also differ in size, in allometry, the relative proportion of their different body parts, in longevity, immunity, physiology, and so on. So whenever you identify a difference between queen and worker, let's say a particular molecular mechanism that differ between the two castes, it's very challenging to actually show that this molecular difference is associated with a specific phenotype and not with other confounding factors. So for this reason, myself and colleagues have been calling for additional approaches. I'm not saying that we should do everything differently, just that we should complement this approach with additional approaches, and we propose to use more flexible systems where we can manipulate the division of labor, and that would provide a more functional approach to the study of the evolution of eusociality in eusocial insects in general and in ants in particular. And in this talk, I will tell you about two such flexible systems. The first one is the clonal rudder ant, Oxley biroi. This species has lost the queen caste, and all basically these ants have reverted to a cyclic lifestyle that is quite analogous to the ancestral cycle. And they give the opportunity to manipulate that cycle and to investigate the mechanisms and factors that could break the cycle and produce a transition from a phasic system to a caste system. Oxley biroi, this model system has been developed by Daniel Kronauer at the Rockefeller University in New York. And the work I will present today is work that has been initiated or conducted when I was a postdoc in his laboratory. And the second system I will tell you about is a more classical ant, the black garden ant, Lysius niger. But I will focus on a particular stage of the life of a colony, and more specifically of the life of a queen, and that's the colony founding stage. And I will try to convince you that this is a very good system to study the emergence of behavioral specialization in ants. And we have been developing this study system for the past 3 years here in my research group at the University of Mainz, Germany. All right, so let's start with the clonal rudder ant. So as I told you, in this species, you don't have queens anymore, and all workers can produce female offspring, other workers, by parthenogenesis. They can clone themselves. There are raider ants, meaning that they raid other ant species, they steal the brood. Here it's a fire ant male pupa, for example, and then they feed their own brood with the brood of other ants. And each colony follows a cycle that is very similar to the ancestral cycle. During the reproductive phase, all the ants are inside the nest, they have developed ovaries, and they lay eggs. And then when these eggs hatch into larvae, the ants regress the ovaries, and they start foraging for food and feeding the brood. And at the end of this brood care phase, when the larvae pupate, all the ants go back inside the nest and they start laying eggs again. And this resembles the ancestral cycle, not only because of the two phases, but also because the cycle is regulated by the presence of larvae. We found that you have a dose-dependent inhibition of reproduction by the larvae in the clonal rudder ant. So here on the y-axis, you have a proxy for ovarian development. On the x-axis, you have increasing larva to worker ratio. And you see that the more larvae you give those ants, the less they reproduce. So because we have this cycle that is similar to the ancestral cycle, or at least that looks similar to the ancestral cycle, and because we have this effect of larvae on reproduction, we wanted to investigate the mechanisms that translate the presence of larvae into reproductive inhibition, and therefore the mechanism that potentially regulate the cycle in the clonal rudder ants. So we came up with a protocol, and I won't go into details, but the idea is that we could get groups of individuals that were exactly the same age, with the same genetic background, they are all clones, but half of them were in the reproductive phase without larvae, and the other half in the brood care phase with larvae. And at this point, we could exchange the brood. We could give larvae to those without larvae and remove larvae to those that have larvae. And this would trigger the transition from one phase to the other and vice versa. And we would collect individuals at different time points, dissect their brains, extract RNA from their brains, and study gene expression changes along the transition from reproduction to brood care and from brood care to reproduction. When we did that, we found a large number of genes that would respond to the manipulation of the brood, and that would basically change during the transition from one phase to the other. We found that the genes involved in one transition were not necessarily the same as the genes involved in the other transitions. And we also found that the time dynamics was pretty different between the two transitions. So when you add larvae, you have very fast changes already after 12 hours, and then the changes are gradual over time. But if you remove larvae, not much happens for the first 2 days, and then after that, you have large changes in gene expression. And this mirrors what we see at the phenotypic level. If you take these ants in the reproductive phase and you add larvae, they inhibit the reproduction right away. But if you take ants in the foraging phase, in the brood care phase, with the larvae and you remove those larvae, it takes them a while before they start developing their ovaries. And in a way that makes sense, that if you have ants in the brood care phase that need to leave the nest to forage for food, you don't want them to start developing the ovaries just because they are away from the nest and therefore away from the larvae. So it makes sense that they are a bit slower to react in that direction. We had all these changes in gene expression. In another study, we wanted to investigate the mechanism that regulate those changes in gene expression, and we tested the effect of an epigenetic mechanism, DNA methylation, and we found that this mechanism is unlikely to regulate those changes in gene expression, mostly because it did not differ between the two phases of the cycle. So from this large list of genes, we could try to identify candidates for the regulation of the effect of larvae on reproduction. And we could look for genes that have a very strong effect size, that maybe were involved in both transitions. And because we had this time course approach, we could also look for genes that were pretty upstream in the changes, that basically changed their expression early after removing or adding the larvae, and that therefore had probably more a regulatory role in the transition. So here I give two examples, insulin-like peptide 2 or ILP2 and vitellogenin. These are genes that belong to the insulin signaling and vitellogenin pathway. I put them here because they show strong changes in both transition, and they belong to pathways that are known to be associated with reproduction in insects in general. And we know these two pathways are connected, and here with the time course approach, that would be more consistent with insulin signaling being upstream of the vitellogenin changes, because we had changes beforehand for ILP2 than for vitellogenin. And this is consistent with what we know in Drosophila, for example. So, okay, this would be a nice candidate for the regulation of reproduction by the larvae, but maybe they're just specific to the clonal rudder ant, and maybe they're specific to their rather unusual lifestyle. And also, well, there is no indication that they would actually play a role in regulating the cycle in the subsocial ancestor of ants. And this was the question that I started with. So we complemented this approach with a cross-species comparison approach. And here the idea is very simple. Probably I make it even more simple here, is that if you have a phylogeny with one common ancestor and you find a trait present across that phylogeny, then it's likely that this trait was already there in the common ancestor to that phylogeny. And we applied this idea to the ant phylogeny. So we used 7 ant species across the ant phylogeny that represented different subfamilies. And we compared brain gene expression between queens and workers, and in some cases, reproductive and non-reproductive individuals. And we found one gene that was consistently overexpressed in reproductive individuals in all 7 ant species, and that was ILP2 that I've just told you about. So to us, the most parsimonious explanation for that is that ILP2 was already more expressed during the reproductive phase when individuals were reproducing in the common ancestor to these ants, and therefore in the subsocial ancestor of all ants. So then that means that if that gene was more expressed in the reproductive phase, it could be involved in the regulation of the cycle, and it could also have been involved in the transition from a phasic system to a Cas system. So to study that in more detail, we went back to the clonal rudder ant. So we knew already that ILP2 differed between the two phases. One problem here is that ILP2 belongs to the insulin signaling pathway, which is known to respond to food. And these ants, they are fed differently between the reproductive phase and the brood care phase. And the queens and the workers that we compared in this cross-species comparison, they also differ in the food that they receive. So we were a bit worried that the pattern of expression we found with ILP2 more expressed in reproductive individuals could have been driven by differences in food intake to which the insulin signaling would have responded. So we repeated the experiment, but this time we controlled for food, and we found in Obiroi that even when food is controlled for, the larvae inhibits the expression of ILP2, meaning that you have a social regulation of a gene, a social regulation of insulin signaling in that species. So the presence of larvae inhibits reproduction, and the presence of larvae inhibits ILP2. So we draw the hypothesis that ILP2 may mediate the effect of larvae on reproduction. And to test that, we synthesized the ILP2 peptide and injected it into the ants. And we found that ants injected with ILP2, would keep on reproducing even in presence of larvae. Therefore, they would not inhibit their own reproduction when larvae are present. That means that by injecting ILP2, we broke the effect of larvae on reproduction. And it's very strong evidence that indeed ILP2 is involved in translating the presence of larvae into reproductive inhibition. But it also means that by doing that, we broke the cycle. We produced ants that would always reproduce irrespective of the presence of larvae. Therefore, we produced ants that were stuck in the reproductive phase. And if you are stuck in a phase, well, it's a good starting point to become a caste. So having variation in ILP2 and therefore variation in how much you reproduce when larvae is present could be one way to go from a phasic system to a caste system. But of course, nobody was there at the time injecting ants with ILP2. So one question is, how could you get variation in ILP2 naturally? And we come back to food again, because it belongs to the insulin signaling pathway. It's likely to respond to food. And because we know queens and workers receive different food during larval development. So one female larva will, when they are very young, they can develop in either a queen or a worker. There are many factors that can affect this alternative development. But the fact that the larva becomes a queen or a worker usually requires different feeding, either in terms of type of food or quantity of food. And this could result in different activation of the insulin signaling pathway and in adults that have different ILP2 levels. So the clonal rudder ants, they don't have queens, but they have what we call intercaste. And these intercastes, they are similar to workers, except that they have more varioles and their morphology is a bit different. Their thorax, for example, is a bit more similar to the thorax of a queen. So they're a bit more queen-like than the regular worker, let's say. And we found that if you manipulate the food that the colonies receive, you get more intercaste in colonies that are better fed, possibly because as larvae, these individuals receive more food. We also found that these intercaste, they reproduce more in presence of the larvae. So they tend to listen a bit less to the presence of larvae by inhibiting their reproduction a bit less. So here we thought, well, maybe that is because they have more ILP2, that they inhibit their reproduction less in presence of larvae. And indeed, looking at the quantity of ILP2 in the brain, we found that intercastes have more ILP2 than regular workers in both phases of the cycle. And that probably means that they have higher baseline ILP2 level, possibly because of differential food that they receive as larvae, and this results in them being more likely to reproduce in presence of larvae. So this is a lot, and I'll try to summarize a bit what I've presented now into this scenario, mechanistic scenario that we proposed for the evolution of queens and workers in ants. So as I told you, the question is, how do you go from a phasic system, from a subsocial ancestor, to a caste system in eusocial insects? But I would say that we first have to think of how you get from a solitary insect that does not do any parental care to a subsocial insect. And we propose in that scenario that the insulin signaling pathway, or maybe ILP2, became responsive to the presence of larvae in that transition. And in a way, it makes sense that to evolve maternal care, you may have food-related pathways, such as the insulin signaling pathway, that would be hijacked by your offspring, so that you basically become hungry because offspring is present, and you forage for food not for yourself, but for the offspring. So if this happened, if ILP2 became responsive to larvae, then you would see this pattern that we assume existed at the time, that in the reproductive phase, when you don't have larvae, you have high ILP2, and then when larvae are present, they inhibit ILP2 and they inhibit reproduction as well, meaning that you are in the brood care phase. So now the question is, how do you go from there to this caste system with high ILP2 in queens, but low ILP2 in workers? Well, as I said, we propose that this might come from differential nutrition as larvae. You can imagine that some larvae receive a bit more food, than others, either at the same time or kind of in a seasonal manner. And those larvae that receive a bit more food would become adults with a bit more ILP2. And these adults with a bit more ILP2 would have a bit of a higher reproductive potential, either by itself or because they would differ in the response to the presence of larvae. And this small difference in reproductive potential, driven by a small difference in larval nutrition, could have been then picked up by natural selection, given, of course, the right ecological conditions, the right benefits and costs, and probably associated with additional mechanisms such as queen pheromones or worker policing and so on. And then all of these could give rise to the queen and worker caste, morphologically differentiated queen and worker caste that we find in ants today. Okay, so that's it for this first part on the use of the clonal rudder ant to study reproductive division of labor and a possible scenario for the evolution of queen and worker caste in ants. I mean, this is a scenario that has been proposed already before, but here we just add a mechanistic layer to it with this ILP2 story. Okay, so now let's move on to the second system that I would like to tell you about. And this is the black garden ant Lasius niger. And here I'm going to talk more about behavioral division of labor, behavioral specialization. So I will focus on one particular part of the life of this ant, of this queen here, and that is the founding stage. And I will try to make the case that it's a good system to study the emergence of behavioral specialization. So queens and workers, when textbook examples of castes, and the main differences are queens do reproduce, but workers don't. Workers do brood care, but queens don't. And this is true in most ant species, but this is true in established colonies, in mature colonies. And the story might be a bit different when you look at the founding stage of species that have independent colony founding. So in this species, usually you have a single queen per nest. And this queen or this colony here, during the reproductive season, they will produce new queens and males. And these new queens and males will fly away. They will mate during a nuptial flight. The males will die. And the new queens will look for a nest site. And there they will shed their wings and start a new colony independently by themselves. So they would lay eggs. And a couple of months later, they will have their first workers. During these 2 months here, they are just one adult with the brood. And because they manage to get workers, it's likely that they actually care for that brood. And this is why I'm interested in this part of the life of those queens and how then they transition to being the queen in a colony. So to study this, we use the black garden ant, Lasius niger. So we use that species because they do independent colony founding, because they are super common in Europe. They do these very large nuptial flights, and maybe, yeah, here in Mainz, for example, we have 2 or 3 large nuptial flights per year. And when you find it in the right location, you find just hundreds of those queens roaming around looking for a nest site. So it's very easy to collect in one day. I think this year and last year, we collected both here more than 1,500 queens in a matter of 1 or 2 days. And then you can put those queens into individual glass tubes, as you can see on the right here. Each tube here has one queen inside. And so you just have water and then cotton. The water is blocked by a cotton ball, and then a queen, and then another piece of cotton. And these founding queens, they don't feed. They don't need to forage for food in that species as well, at least. So you don't need to do anything. You just put those tubes in the dark at 21 degrees. And then 2 months later, you get a small colony. But we did monitor a bit what was happening during those 2 months. So here we followed 24 queens after the nuptial flight in their individual tubes. And right away, in the first week, 3 of them died. But after that, the 21 that remained, they were alive until the end of this monitoring, and they may still be alive today in the climate chamber downstairs. So what we see is that first they lay a lot of eggs, and then you have this drop in egg number when the larvae emerge, which is expected because the larvae emerge from eggs. But also you see that the drop in egg number is larger than the increase in larva number. And that's probably because those larvae, they eat the eggs that are around them to start growing. And we have seen such cannibalistic behavior. And we have observed it in the tubes as well. And then from those larvae, some of them became pupae. And from those pupae emerged some workers. Those 21 queens that survived, all of them produce workers after 60, 70 days. So they are quite good at doing that. And the question is, well, do they do that by actively caring for the brood? And then do they stop caring for the brood once they have workers? So to address this question, we simply observed some of those queens before worker emergence and then after worker emergence. And we found indeed that before worker emergence, founding queens do a lot of nursing behavior, and then they do less or even no nursing behavior at all after worker emergence. So you do have an association between the emergence of workers and the queen behavior. And it suggests that the social context affects the behavior of those queens. However, founding and established queens differ in several aspects. Those founding queens, sure, they don't have workers, but also they are not fed, and they are younger by definition. And the established queens, they do have workers, but they are fed and they are older. So we wanted to disentangle these different factors to really find out which factor was responsible for the queen behavioral specialization. And that was actually a bit tricky to do, because what you want to do if you want to address that question is to manipulate the presence of workers. The problem is that either you wait for those queens to have workers, but then it's too late, or you take workers from another colony, but then something really bad happens. The problem is that ants communicate via chemicals, and they have on their body a blend of odors that we call cuticular hydrocarbons. And this blend of odors is specific to the colony they come from. So if you take workers and you give them to one queen, the queen will know right away that these are foreign workers. And those queens are super aggressive. They will just cut the workers in half in a matter of seconds. And the workers could also be aggressive toward the queen. So you cannot simply add workers that come from other colonies, and you cannot use the workers from those queens. So we developed a protocol, and the idea is we go in the field, we collect a lot of pupae of those cocoons here, so that are basically, that will produce, from which workers will emerge very soon. And we collect hundreds and hundreds of those pupae that we put in laboratory nests with some workers to take care of them. We don't feed those experimental colonies. And then in the morning, when we want to do the experiment, we go through those colonies and we catch what we call callow workers. These are workers that were born in the past 4-6 hours. And we can find them because their color is super light. They are light gray. They don't have the final color yet. So they're easy to pick up. And very importantly, these callow workers, they don't have the colony odor yet. So they don't have this blend of odors on their cuticle. So when we give them to the queen, they are accepted by the queen. And we found that they actually change the behavior of the queen as if they were the workers of that particular queen. So we had then this experiment here where we use same age queens, and we either gave them callow workers or not. And in a full factorial design, we either fed them or not. Feeding is a bit tricky as well, because as I said, they don't forage for food. They're not big fan of food at this stage in their life. So, but if you're very patient and you use a tiny spoon with a mix of eggs and honey, and you approach it to the mouth of those queens, they will very nicely eat that food. So we did that every day for 7 days, and all of these days we recorded the behavior of those queens as well. And when I say 7 days, it's 7 days after we added the calories or not. And we found that even though all those queens were the same age, we did find a strong reduction in nursing behavior in presence of workers, and irrespective of the presence of food, either as the main effect or as an interaction. So that means that the presence of workers is necessary and sufficient to change the behavior of the queen. So you have a social control of the behavioral specialization of those queens. Before they have workers, these queens are pluripotent, they both reproduce and they care for their young, but as soon as their social context changes, then they become specialized in being a true queen. So from there, we wanted to try to understand a bit more the effect of the social context. So we asked a series of a few questions. The first one is the queen behavioral transition reversible? And what we did was to give workers to queen, but then we remove those workers after 3 days. So here on the left side here, we have queens without workers, queens with workers do less nursing behavior. And then we have this category that had workers for 3 days. At the end of the 3 days, they were super low, similar to the green dot here. But then 24 hours after removal of the workers, they already went back to doing nursing behavior. So they reverted to doing nursing behavior. And every year, we collect those queens, we do experiments with them, but we also keep some of them that establish colonies in the lab so that we have lab colonies of different ages. And here we used colonies that were 2.5 years old, and we removed all the workers from those colonies. So those queens had workers for 2.5 years old. They had not been doing any nursing behavior for 2.5 years. And we found that 24 hours after you remove the workers, they go back to doing nursing behavior as the founding queens. So you do have this strong reversibility of the queen behavioral transition. So yes, these two experiments show that the queens can revert to doing nursing behavior after the removal of workers, even if they had workers for quite some time. To me, this is kind of an unexpected flexibility that they can go back to doing this nursing behavior, because we tend to think of ant societies as very fixed because of this caste system. But despite this caste system, the social context is super important to regulate the behavior that they are doing. So here we find that the queen behavioral specialization is initiated by the presence of workers, but it's also constantly maintained by the presence of workers. So we have a constant importance of the social context for the behavioral division of labor to exist in this species, at least. And it reminds me a bit of the effect of queen presence on worker reproduction. So in many ant species, if you remove the queen, the workers will develop their ovaries and sometimes lay haploid eggs that will become males, but they start being reproductively active. And it's kind of the same, that basically you have a social control of reproductive division of labor. It's the presence of the queen that prevents the workers from reproducing. Probably not, I mean, probably that the worker inhibit their own reproduction in presence of the queen, should frame it that way. But if you remove the queen, then this goes away and the reproductive division of labor is not as clear-cut. And we have a similar effect at the behavioral level with this constant social control of the queen behavioral specialization. And on a side note, maybe this gives some information on the mechanisms that are necessary to maintain the ability to do brood care, or the mechanisms that regulate brood care, because that means that maintaining the ability to do brood care is probably not very costly, because it's very unlikely in a natural situation that these queens have to do brood care again in their life. I mean, in this species, you have 10, 20,000 workers. So it's very unlikely that all workers die suddenly and the queen have to revert to doing brood care. So if it was costly to keep that ability, they would probably not keep it. So I think it some kind of indications that whatever mechanism are required to maintain the ability is not costly. Another question we asked was, okay, so far we've been comparing queens with and without workers. But we can ask the question whether we have a dose-dependent effect of the workers or a gradual effect of the workers. So once you have workers, if you have more and more workers, is the queen doing less and less nursing behavior? Or on the contrary, is it some kind of an on-off mechanism? Workers are there, I stop doing nursing behavior. So we use queens, founding queens again, and callow workers, same setup. This time we had a queen with 15 larvae, and we either gave them no workers or 1, 2, 3, or 5 workers. And we found this non-negative, non-linear negative relationship between nursing behavior and the number of workers. But it looks like this decrease here is mostly driven by the first category with no workers at all. So we repeated the same analysis by removing this category. And here we were not able to detect the effect anymore, even though there was still a tendency for a decrease over time. We did the same now with 5 larvae and not 15 larvae, and we found the same pattern. Clear effect with the category with 0 worker, not able to detect that effect without that category. So here I don't want to make too much of a strong conclusion because it's a bit difficult to differentiate between the two just from this data. But what I think is for sure is that most of the decrease in nursing behavior is driven by having only 1 worker. And there might be additional decrease, but at least we were not able to detect it in this experiment. And in that case, for example, this was mostly driven by this 1 queen here. If you remove it, basically the relationship is almost flat. So this is one line of evidence here that maybe it's more toward an on-off mechanism. And the other line of evidence is that the pattern was the same with 15 larvae and with 5 larvae. So the idea is, if it's a dose-dependent effect, it should not necessarily be because of absolute number of workers, but rather a relative number of workers relative to the amount of work that has to be done. So that's the idea, that you have more workers, you have less nursing tasks to be done, so the queen is doing less and less nursing tasks. So the fact that we find the same pattern irrespective of the amount of work to be done, 15 larvae or 5 larvae, to me again points more to the direction of an on-off mechanism. And consistent with that, we again did the same experiment with established queens. This time they were 3.5 years old. We removed all the workers but 5. And then we looked at their behavior. None of those queens did nursing behavior. Then we remove another worker in the morning, look at the behavior in the afternoon. None of those queens did some nursing behavior. With 3 workers, none of those queens. With 2 and 1 worker, one of those queens was doing some nursing behavior, but we cannot be sure that this difference between 0 and 1 is not expected by chance. And finally, when we went from 1 worker to 0 worker, then 11 out of the 24 queens started to do nursing behavior. So this is also more consistent with an on-off mechanism. That it's not necessarily how many workers are there, but just the presence of worker that is important for the queen to change her behavior. And in a way that could be a bit surprising, because then if 1 worker is not enough to take care of the brood that is present, then not taking care of that brood could be costly. But I think we have to put that into the context of this founding stage, where basically 1 worker in that context may be sufficient to care for the number of larvae that are present, which is not a lot. And also maybe 1 worker anyway announces more to come in the few next hours or the next day. And therefore, in the context of founding stage, where basically the change in queen behavior, that's in that context that this change in behavior has been selected for, then maybe 1 worker is enough to care for the larvae, or means that it will soon be enough to care for the larvae. So that means that changing your behavior in that context with only 1 worker might not be costly if the larvae are fine. You know, if 1 worker can take care of all the larvae and they all survive and they all develop properly, then maybe it's not costly. And then this response, this on-off response from the queen could have been favored and selected for if it provided some benefits to the queen. And those queens can then focus on reproduction earlier if they don't wait to have more workers. And maybe they lay more eggs and their colonies grow faster. And then they would find a benefit of having this very fast on-off response to the presence of workers. So far, yes. And the third question that we wanted to ask about this effect of the social context is, well, how do workers communicate their presence? And I told you that workers, ants in general, communicate via chemicals and via this layer of cuticular hydrocarbons that they have on their body, these CHCs. So we asked the question, well, if we just give the odor of workers, is it sufficient to affect the behavior of the queen? Is it the signal that the queen is using to change her behavior? So we extracted CHC from workers, gave this CHC to the queen, and we did not find any reduction in nursing behavior in response to the odor of workers. So the odor of workers is not sufficient to affect the behavior of the queen. Then we thought, well, maybe the queen needs real worker to be there. So we wanted to find a way to have workers that are present, but are non-functional, are not caring for the brood. So we gave workers to queens, but we separated the workers from the queen and the brood with a wire mesh, so that the queens could still alternate with the workers. They are still aware of their presence, but the workers cannot care for the brood. And there again, we found that when workers are separated by a wire mesh, there is no reduction in the nursing behavior expressed by the queen. So that means that it's not enough to produce the queen behavioral specialization. So what I would say here is that for the workers to have an effect may require closer contact between the workers and the queen, like actual very close interaction between them. Or maybe you need the workers to actually care for the brood. Maybe you need the brood to change their signal that they need something because the workers are now there interacting with them, and then only the queen will stop doing the nursing behavior. But I think here we need to work a bit more to understand how the presence of workers is communicated. So far I've been talking about Lasius niger, and it's a single species, and it's around the context of colony funding where queens do nursing behavior anyway. But because we found this result that even established queens can go back to doing nursing behavior in that species, well, it raised the question whether it might also be the case in other species. So we have another species in the lab, Terminothorax nelanderi. It's very easy to collect in the forest close to Mainz. These have small colony size. They live in acorns or small twigs. They have dozens of workers on the queen. And so you can collect established colonies right away in the field. And we used 18 of those established colonies, manipulated the social context, and we found that for 8 of the 18 colonies, the queens went back to doing nursing behavior at some point. So you also saw this ability for queens to go back to doing nursing behavior in that species. And there was another paper published this year on the little fire ant, where they also manipulated the social context. They removed half the workers. So it's not all the workers, just half the workers here. And they found that many of the queens here went back to doing nursing behavior. Actually, those queens would also forage for food and so on. This species is an invasive species with, let's say, an unusual reproduction mode. And the authors put this result more in the context of that basically this would give them an advantage as an invasive species. But I think it can also inform us on the general ability of queens to do nursing behavior. And what's interesting in these two species belong to the ant subfamily Myrmicinae, which is a different subfamily from Lasius niger, which is a Formicinae, and they have been apart for a very, very long time. So maybe this ability of queens to do nursing behavior is somehow reminiscent of the ancestral behavior, and it's conserved in these ants that have been apart for quite some time. So assuming this, that basically what we see during the funding stage and also later in the life of a colony, this ability to nurse a brood, assuming that it's ancestral, it may be similar to how the nursing behavior was regulated and controlled in the subsocial ancestor of ants. So it led us to ask the question, well, those founding queens, are they analogous to the subsocial ancestor? And if we go back to the difference between queens and workers, we have this difference in brood care and reproduction. But if now we take what we learned from the first part of this talk and we apply it here, when I say reproduction, I actually mean inhibition of reproduction by larvae. And this is something that we know from several ant species, that if you remove the queens, the workers will start to develop their ovaries, as I told you, but they would do so less in presence of larvae compared to in absence of larvae. So larvae inhibit reproduction in workers, and larvae do not inhibit reproduction in queens. Usually, the more larvae you have, the more eggs are produced by the queens. So now, if we put that back into the context of this comparison to the subsocial ancestor, it would look like this. When we look at brood care, we know queens don't do brood care, but the subsocial ancestor used to do brood care, and our founding queens, they do brood care. Now, when we look at the inhibition of reproduction by larvae, we know it doesn't exist in established queens, and we know the subsocial cycle of the ancestor was regulated by the presence of larvae via what we know about ants, but also via what we know about closely related wasp species to the subsocial ancestor that are still subsocial nowadays, and that have a similar cycle that is also regulated by the presence of larvae. So the larvae inhibit reproduction in that situation as well. So we asked this question for the founding queens. We took founding queens either before they have workers or after they have workers. We isolated them with or without larvae, and we counted how many eggs were found in the box after 5 days. And we found that for founding queens, the presence of larvae reduced the number of eggs in the box, but for established queens, the presence of larvae increased the number of eggs found in the box. This is consistent with an inhibition of reproduction for founding queens, but not in established queens. So again, we would have founding queens that are more similar to the subsocial ancestor, both in terms of brood care and in terms of reproduction, than they are to established queens, even though those founding queens, they will be established queens just a few weeks later. So because we have this similarity, and because we find this ability to nurse the brood to be present in young and older queens and maybe in different species, all of that leads us to propose that maybe this transition from founding to established queens may inform us on the transition from subsociality to eusociality, because we have this transition from a pluripotent individual, the founding queens or the subsocial ancestor, to a specialized individuals, the established queens or the caste system. So of course, we're talking about way different things because it's within the lifespan of an individual in one case, over evolutionary time in the other case, but that might be one way to approach the question that might be a bit complementary to what has been done so far. So for that reason, we are interested in the mechanism that translates the social context into behavioral specialization. And I don't have time to go too much into details. And also this is ongoing work and I don't have much to say for the moment. But what I can tell you is that we are using RNA-seq to compare brain gene expression data between queens without workers and queens with workers. And we have already identified candidate genes that differ between queens with and without workers, and therefore queens that do or don't do nursing behavior. We are developing qPCR primers for those candidates, as well as an RNAi protocol to downregulate the expression of those candidates and see the effect on the behavior of the queen. We're also interested in the molecular mechanism that could regulate those changes in gene expression. And most specifically, we're interested in epigenetic processes. We use a very simple approach for the moment, is to use chemical inhibitors that basically inhibit specific epigenetic processes. And we can treat the queens with these inhibitors and see whether that affects their behavior and more specifically their response to the presence of larvae. And of course, the idea for all these experiments where we try to manipulate the molecular mechanism is to see whether we can experimentally produce queens that do not specialize in response to the presence of workers. So queens that would keep on doing nursing behavior even when workers are present. So queens that do not become true queens, and basically that would be colonies where you don't really have behavioral division of labor because the queens would still care for the brood. And that would be interesting per se to understand the queen behavioral specialization in Leisure Niger. That could potentially provide ideas of how you go from this pluripotency to this specialized system in general in ants. And also then we would get maybe the opportunity to study a bit the benefits and the cost of this specialization, because then we could have colonies where you have behavioral specialization and colonies where you don't have behavioral specialization of the queens. All right, so that's it for the second part. I would like to briefly conclude the talk by giving two take-home messages. The first one is about the social context. So we found, using the clonal radar ants and also a cross-comparison approach, that variation in response to the social context is central to the evolution of reproductive castes in ants. We also find that the social context drives the emergence and the maintenance of behavioral specialization in Leisure Niger and maybe in other species of ants. Another way to say it is that queens are only queens once they have workers. The second take-home message is about flexibility. These ant societies, they are typically seen as very derived, very fixed system where you have these different castes that fulfill different functions and they are bound to performing these functions. But actually, we see here that just manipulating the social context show quite some flexibility, maybe more than expected. That you can take queens that have been specialized for years in not caring for the brood and only doing egg production. And when you manipulate the social context, then they go back to nursing the brood. So I think it's important to take that flexibility into account when studying social insects in general. And finally, what I have presented today could not have been done without using these flexible model systems where you can actually experimentally manipulate division of labor. And I think it shows the power of using flexible system. It's important to use such system in addition to other approaches to study the evolution of insect societies. And with that, I would like to thank all the collaborators and students that participated in those projects and collected the data. These are the OCRI people at the Rockefeller University that used to be there at the time or are still there now. And of course, most of all, Daniel Kronauer there, who has developed the clonal weather hand as a model system and is doing very nice work with that species. And I would also like to thank the laziest people here at the University of Mainz. These are students in my group. So we have several PhD students that contributed to this project, even though it's not the project of any of them, but they were happy enough to have this as a side project and to collaborate on this kind of central project in the group. And there are several master and bachelor students that collected the data, did the behavioral analysis, and basically provided all the content necessary to build the stories that I've told you about today. And I would also like to thank the Department of Behavioral Ecology and Social Evolution here at the University of Mainz. We are 4 groups working on social evolution using ants and bees. And there is really a nice atmosphere, a lot of brainstorming, exchange of ideas, criticism. It's very dynamic. And I think it really contributes to good science. And if in the audience there are any students interested in doing an internship or applying for a PhD or a postdoc in our department, feel free to check out the webpage of our groups, not only mine, but also of the other PIs, and to contact us if you have any question. I would like to thank again the organizers for this terrific job that they have been doing with the seminar series, and I thank you for your attention.
Yes, thank you, Romain, for a fascinating talk. It's great how you can go in such molecular detail and be able to do experiments. Now we are at the discussion. You can ask questions. As a reminder, if you have a question, simply type a question mark into the chat function, and then we will call you up. And also, if it's your time, please, even though we know most of you now, quickly say your name and then where you're from. For the people on YouTube, if there are any questions, you can also put it there in the chat function, and at some stage I will ask the question. There's a time lag between YouTube and Zoom, so you might have to wait some time. So the first question is coming from Michael Taborski.
Yeah, thank you very much, Romain. Extremely interesting work. I think this flexibility of ants, especially your clonal radar ant, but also other species, opens up the possibility to check in even eusocial insects for the important benefit of division of labor for the success of societies. I think this is a great opportunity, and I'm not sure whether, I mean, you were not talking about that, but I'm not sure whether there are any data that allow to compare, I mean, highly specialized and less specialized species, or for example, your clonal radar ant with related species with regard to their ecological success, because I think this is a question of very general interest. To understand the importance of the division of labor for the evolution of advanced levels of sociality.
Yeah, that's a good point. It's a good question. So I guess we could see it in different ways. What you mean probably is to be able to compare, in the case of the clonal radar ants, maybe closely related species that would not have lost the queen caste, for example, and then being able to have some kind of a comparison between a caste system and a system that went back to this cycle. I mean, in theory, it's feasible. We know of closely related species that do have queens, but we're always getting to the issue that to assess costs and benefits, lab work might make it a bit more restrictive, because then it would not necessarily recapitulate the proper ecological conditions or things like this. And for that species, it's a bit tricky because they only live underground, so they never go outside. So it's very difficult to observe them or do anything with them in the field. And probably then the closely related species would be quite different in many aspects compared to that particular species. I know what Daniel has been doing is to try to find if there were populations, because they are invasive species and these have been collected where they are invasive, but to go back to their area of origin and try to find if there are populations that still have the queen. Because then you would even have the same species, but where you could compare populations with queen and without queens. And I would, I guess, exactly answer your question. I don't know if he has been successful in that now, but at the time, they could not find populations with queens, mostly because it was also difficulty to access this area of origin and just to find more of those ants. For the other, like, more established species where you can manipulate a bit this division of labor, that's a bit what I try to hint at by saying that we could, if we could manipulate from a molecular perspective the ability of queens to switch to not care for the brood anymore and to have queens that always care for the brood, then in the lab we could end up with colonies with a queen that still care for the brood, and then maybe then can less invest into egg production, and compare them to colonies where the queen can actually focus more on producing eggs and with a stronger division of labor. But then, of course, if you do experimental manipulation to get those queens, we would have to be aware of confounding factors of toxicity of treatment, or whatever, anything else that we could affect as well. So I agree with you that it's something very important, and I think again that work in the lab of Daniel Kronauer done by Yuko Ulrich also on the Kronauer ants, where she manipulated the size of the colony. And if I remember correctly, this had effect on fitness correlates. And I think they put that in the context of more or less efficient division of labor depending on colony size. So that could maybe link to your question a bit as well. I don't know if this answered, it was a bit, several things all over the place, but this is what you made me think of when you asked your question.
Yeah, thank you very much. It was a good answer. I think it shows that there is a lot of potential for future studies also in that direction. Thank you very much.
Thanks.
So the next question is by Sari van Bell.
Hello, I'm Sari van Bell. I'm at the University of Texas in Austin. Yeah, you hear me? Yeah.
Yes.
Talking about colony size and your lab setup, in that you mentioned that of the Lasius niger, that those colonies in their natural environment are something like 20,000 workers. Yet you said at one point in your lab you have 2.5-year-old colonies from which you removed all workers, which I kind of think is an extremely daunting task if it is 2,000 or 20,000 workers. So I'm a little bit like, well, how big are these colonies we are talking about?
Yeah, no, they were not that large. So in the lab, the colonies don't grow as large as in the field. So these colonies were probably a few hundred workers max. So it was not that difficult to remove the workers. And it's a good point you raised. And what we're trying to do is to do what you say would be very difficult to do, which is to use mature colonies from the field that have closer to this number of workers that we would expect, and to do the same experiment. And so basically, I would not remove all the workers. I would simply put the queen aside, maybe first with a few workers and then remove them one by one. The problem is that getting field colonies with the queen is super challenging. They are often very deep. And so this year, I think we collected 2 field colonies where we managed to get the queens. I have colleagues that are trying to do that as well. But indeed, that could affect the result. It would be interesting to see whether we still find this ability to reverse during the nursing behavior when they have so many workers. Yes.
Thank you. I really liked your talk. Very interesting.
Thanks.
So then the next question is by John Strassman, please.
Hello, I'm at Washington University in
Hey.
St. Louis. You hear me? Yeah.
Yes, I hear you. Hey, John.
Hey, so I thought that was just a fantastic talk, but of course I love social insects. And I just think the things you can do with ants are completely amazing. So this is just, I don't know, maybe a bit of a quibble, and that is that you motivated your fabulous work by talking about origins of sociality and subsocial species. And I just think you ought to lose that because ants are just too far away. We have species of bees. We have Lasioglossum species where you have the transition to eusociality within the same species. And it just seems like you can do so much more with the wasps and bees that actually have the transition. You know, unless you can show that the mechanisms that you're seeing really are the same in the primitively social wasps and bees, you know, I just don't think you need to have that framework.
Yeah, I completely agree with that comment. What I could say is, well, first that it could be seen as different complementary approach to study this. And what we know from previous work recently is that it's likely that these different origin of eusociality, they went through different routes, especially when we talk about mechanisms. So you have now several papers that struggle to find a common mechanism in even the different origins of eusociality in bees. So you could make the argument that each of those origins of eusociality are interested to look at by themselves, and that it might be very difficult to find a common pattern, a common or genetic toolkit for the evolution of eusociality. Assuming this, then looking, if you want to understand how the diversity in ants evolve and how ants evolve, you have to stick with ants, because whatever you would find in bees and wasps, it would be super interesting, but would not necessarily apply to ants. And but I agree with you that there are very good system for that. You're right, facultatively eusocial bees, and also wasps that probably would be closely related to this subsocial ancestor. We could gain a lot from that, and I think a lot of people are working on this as well. But I just take it as different approaches to the question. And I do agree that ants are super derived. That's kind of their characteristics. But by finding such flexibility that comes back, that exists, finding that they go back to this cyclic lifestyle super easily if they lose the queen cast, maybe what mattered at the time is still there somewhere, and we could assume that maybe there are mechanisms that regulate it that were already present in this subsocial ancestor. And I think the cross-species comparison gives you an idea of that even though we use derived species because they all share something, it's likely that this was already there at the time. So I do agree with you that what I've told you about does not apply to the origins of eusociality, but applies to the origin of eusociality in ants. And that indeed, then we cannot make it as general as maybe I presented it out of simplicity. I agree with that, yes.
Well, great talk, thank you.
Thanks.
And the next question is by Stephen Trumbo, please.
Hi, this is Steve Trumbo from University of Connecticut. And that was a great talk, a fantastic talk. I had a question of, is any of this work integrated with a study of juvenile hormone, which obviously is involved in reproduction in lots of insects and responds to nutritional signals, differs between subsocial and especially the highly eusocial insects have some different relationships with JH, but has there been any work that you know of or maybe could be done with integrating JH into this?
Yeah, definitely. It's a pathway that pops up in our gene expression analysis, like that we are currently doing with the LASIOS data set. And I've been interested in JH as well for quite some times. And we worked quite a bit during my PhD with Laurent Keller on the effect of juvenile hormone on cast determination. And we are currently doing the kind of manipulation experiments that I was telling you about with juvenile hormone treatment, or at least treatment with metoprine here, and also some inhibitors of juvenile hormone. So it's very early steps, just trying to see again if we treat the queen with an inhibitor of the juvenile hormone pathway or with a synthetic analog of juvenile hormone, do we find that this affects their ability to switch their behavior in response to the social context? I think the student that is the master student is actually watching us right now. So she has done the experiments. We have the long hours of videos of the queen, and the behavior analysis still remain to be done. But yes, we're starting to look into that as well. It's a very good point. And insulin signaling, vitellogenin, of course, there is the juvenile hormone pathway somewhere there because they're all interconnected. Yes.
Thank you.
Thanks.
Then I have two questions from YouTube from Marco Pracic. I will ask them one by one because I want to extend on the second question if I have the similar question or related to this. So he says, fascinating research, Romain. Two questions. First, why were you surprised that queens are flexible in nursing behavior?
Oh, maybe I was naive, but because I've never seen a queen do nursing behavior before, apart in the context of the funding stage. I think from many species I've been working with, and especially as a PhD student and as a postdoc, spending a lot of time looking at ants for many different reasons. Maybe I was unlucky, but I never saw the queens interacting with the brood. And the other reason is really because of this textbook approach that queens and workers do different jobs, and that's it. But again, maybe it's not that unexpected now that I think about it, because queens and workers in most species, they develop from the same genetic background. So one female egg could give rise to a queen or a worker. So any difference between the two is due to differential gene expression, and the building blocks are the same. So as adults, they still have the ability to do whatever the workers do. So it's not that surprising that they do nursing behavior in some conditions. But I just wasn't expecting to see it in the context of more established queens. And also, another point is that you have these papers that describe morphological changes in the brain of founding queens, but that's mostly before they fly out for the nuptial flight compared to after they have done the nuptial flight. And basically the part of the brains that are important for them to fly or to use visual cues during flying degenerate, and there is a rearrangement of the brain morphology, basically. And I was thinking that maybe something similar could happen when they lose the ability to nurse the brood, that whatever part of the brain they would use for that behavior, because they don't need it anymore, it would just degrade. And if that was true, then you would expect that they could not go back to doing nursing behavior. But it was not based on anything, it was pure speculation.
Yes, then he asked, extending on this, second, some queens did not restart nursing behavior in your experiment. Do you expect they would do it if they had more time, or some queens are not flexible? And this is also where I had a question, might it be kind of related to personality? I took a screenshot here of your graphs, I remember. So when you have this decline from 1 or increase of number of workers from 1, 2, 3, 4, you didn't find a significant relationship because there was variation between individuals. If you would have done that, what you did in the next experiment where you did sequential addition, if you would do with the same, do you think if you would do with the same queens sequential addition of workers, that you might find an effect like this is maybe a queen with a different personality than this one, and they might find in this one it would decrease? And you think there might be something like personality playing a role?
That's a good idea. We do know that there is individual and colony level personality in social insects, so that wouldn't be surprising to find these. I think I remember a paper that compared the likelihood of queens to care for the brood and the likelihood of their first cohort of workers later on to care for the brood as well. And that there was variation among queens in the founding stage, and this basically correlated with the variation among workers that were produced by that queen. So it would be kind of heritable behavioral differences here. I agree with the variation point. That's for sure that we do see variation across queens. And we also have experiments, I haven't shown them here today, but where we never have workers. We just have queens with different numbers of larvae. We just wanted to see if queens would do more or less behavior, nursing behavior, depending on how many larvae they have. And within a group, so like, let's say we have 24 replicates for the group with 10 larvae, within that group, we would see a lot of variation across queens in how much nursing behavior they would do. But we had different time points, 24 hour, 48, 72. And what we find is this variation was not necessarily consistent over time. So the queens that were really high at 24 hours were not necessarily the high ones later on. We didn't find a lot of repeatability in nursing behavior across the time points. But this is not, I mean, we didn't do that experiment for this question, so possibly it would be possible to design it better. And even if it was not the case, it would still be interesting to understand why we have this variation then. It's just noise, or is it just rhythmicity that is differentially synchronized between the queens? And it's a good question. I I completely agree that there is variation across those queens. I'm not sure where it comes from.
Okay, thank you. And the next question is from Facundo.
Hi, Facundo.
Hey.
Really wonderful talk. Thank you.
Thanks.
Just thinking about the genomic mechanisms, I was wondering if you can hybridize some of these ant species, and if so, to what extent? And if not, can you cross-foster some of these like pheromone neutral or larvae to other colonies or other species?
So the larvae, you can move them across colonies. That's not too much of a problem, sometimes even across species. But yes, I'm not really sure why we would use that for, but I can already answer to your other question in terms of probably genetic engineering and the ability to actually manipulate those queens. So there is a big limitation, is that only the queens will produce the next generation. So you could imagine trying to develop genetic engineering tools like CRISPR-Cas9. It's already very unlikely that you get the particular mutation you want. But here in social insect, we would have the extra hurdle of it should be in a queen and not in a worker, so that can go to the next generation. And those queens, the lasers, for example, they don't mate in the lab. So you couldn't get a next generation in the lab. However, in the clonal radar ant, Daniel Kronauer at Rockefeller developed a functioning CRISPR-Cas9 protocol. And he has now developed basically several mutant lines, because then once you get the mutation you want, you can just expand the line because they reproduce by parthenogenesis. So he has several mutant lines corresponding to several mutants, and he has been super successful at doing that in his lab. But with typical ants, we still have quite some hurdles that are difficult to deal with, I would say, for actual genetic engineering. Yes.
Okay, wonderful. Thank you. I'll have to check out that work.
Yeah, thanks.
So the next question is by David Queller, please.
Hi, David Queller, Washington University in St. Louis. Really nice talk. I particularly like the part about trait loss. I've become really interested in trait loss, especially in the context of major transitions, because it seems like the essence of it, queens lose some traits, workers lose other traits or abilities, and at some point you reach the stage where they're completely dependent on each other, sort of at Wilson's point of no return. So it's really interesting that you look for the possibility of the loss of the trait of established queens nursing their young and failed to find it, even though I think you were saying that they probably almost never need that trait in nature. Maybe I was reading too much into it, but
no, I think so. I really think so. First of all, I think it would be very unlikely to lose all their workers. I think I don't see a lot of situations where they would lose all their workers. I mean, then the queen would die as well. If there was an event that would kill 20,000 individuals, probably the one that is also there would die as well. And even if there was a major mortality event, I would still assume that there are at least a few workers, for example, nurses that are close to the queen anyway, that would still be there. Yeah. But I mean, it's kind of more speculation, but yeah, I tend to think that I don't see a situation where a queen would go back to being by herself in mature colonies. And even if it happens, even though she could still care for the brood, she needs the workers to be fed. And I'm not sure she would anyway survive to basically get the benefit of being able to care for the brood.
So then the question becomes why they don't lose that trait when they lose many others. And maybe it's just that it's tied too closely with the ability to nurse early in the colony cycle. You can't lose one without losing the other. So the end of my question is, has anybody looked at this trait in ants with budding reproduction such that the queen never has to do this behavior? Does she lose the ability to nurse young entirely?
Yes, very good question. We did that. And I would keep it so far as anecdotal because we did only one experiment, but basically the same idea. We used the Argentine ant. that is highly polygynous, that reproduce by fission. So for those of you who don't know, that's basically that in a colony, you have many queens, and sometimes some queens leave with some workers and they start a new nest. So you never have a queen by yourself founding a new colony and therefore doing nursing behavior. We took 24 of those queens from these large colonies that we have here in the lab, isolated them without workers but with brood, and we didn't see, we saw one event of nursing behavior out of the 24 queens and 3 hours of videos per queen. So I assume it might just have been an accidental interaction, let's say, but definitely not to the level of what we see for lasius. And the temnothorax ants, the other species, they also do independent colony founding, the other ones that we found some nursing behavior. So I would say it goes in the direction of what you say, that it might be because they need to keep that ability during the founding stage, that they are still able to do it later on. But in species that don't need to do it at any point in their lifespan, they may have lost the ability to do it over evolutionary times. But the goal is to do it with other polygynous species that do fission to really, it's always the same, showing that something doesn't happen. It's always a bit tricky. But yeah, it's a very good question. It's something that I'm really interested in knowing the answer, I guess.
Thanks.
And I myself have another question, which is a very basic question. I'm showing again how little I know about social insects, and you might even have said this in your talk. With regards to your experiments with the clonal radar ant and the insulin-like peptide 2 and related to social evolution, I assume the clonal radar ant, it's a clonal species, so all females are genetically kind of identical. And then my question is whether first this is correct, and second whether one can assume that this was an ancestral state before eusociality, or whether it's rather a derived state, and then the physiological mechanism might also be a derived mechanism. It might be more difficult from there to make conclusions about the way social evolution went.
So yes, they reproduce parthenogenetically, they clone themselves. Yes, it's a derived system, so they used to have a queens and to not reproduce parthenogenetically. So it's indeed associated with the loss of the queen caste. I don't know. We don't know whether it was in, yeah, it's correlated with it at least. And yes, that was one of the worry that we had when we did these experiments, is that whatever we see might not, for example, ILP2 being different between the two phases, might be specific to that species and their mode of reproduction or anything else. And that's why it was reassuring to see that we would identify the same genes when doing the cross-species comparison, to show that it was not specific to that particular ant, but that we could find that pattern across the phylogeny of ants. But yes, it's a very derived system.
So if one doesn't look at the physiological mechanisms, what are the ideas in the ant biology of how eusociality evolved? What was the subsocial species before the eusocial species? What is the idea that it was like in wasps, closely related females having a nest together? What is the idea here that people have?
Yeah, I mean, it's different possibly different in different origins of eusociality. I mean, this has there's been quite a lot of debate about that. So I would say that one first very big difference is the difference between termites and the eusocial Hymenoptera. What is commonly accepted, that is, in termites, what evolved first is kind of a soldier phenotype, in the sense that defending the nest was the first step toward cooperating, while in eusocial Hymenoptera, the first step was to care for the brood. And that might be because in termites, these are hemimetabolous insects, so the brood can take care of themselves, basically. So you don't need this brood care stage, as in for eusocial Hymenoptera. And now there have been a lot of discussion in Eusocial Hymenoptera, a lot of hypotheses, and I would probably be wrong if I was trying to give too much details. But I think what is commonly accepted now is that eusociality evolved repeatedly, and when it evolved in Eusocial Hymenoptera, that was in cases where you had probably a monoandrous, monogynous system, so one mother that mated with one male. And that's basically some daughters staying at the nest to help the mother, and then led to kind of cooperation across those individuals, among those individuals. But there have also been proposition of different routes. I don't remember the name of the route, but where it would go through aggregation of individuals that then would cooperate. But I think there is more debate in whether or not that is consistent with the data that we have today. I'm pretty sure other people in the audience, like David or John, would have very good idea or very good insight into that. But it's kind of a complex topic, yes.
So the change of reproductive to carrying phase is not generally believed to be the ancestral form.
I think it's commonly accepted since the work of Marie Jane West-Eberhardt in WASP, and that we see this subsocial cycle also in many subsocial WASP, that this is a likely route for the evolution of eusociality in ants. In ants, there was a single point of single origin of eusociality. In bees and wasps, you had several ones, and there I know less about whether there have been different routes have been proposed within those lineages, or whether it's also assumed that it was through this kind of a subsocial ancestor that first would do brood care and then would evolve further cooperation.
Okay, thank you. And we have a question by Suleima.
Yes, hi, Suleima Tang Martinez, University of Missouri in St. Louis.
Hey.
Actually, can you hear me okay?
Yes.
Okay, so I initially had two questions, but Karsten just asked exactly what my first question was going to be, which was about whether, we're talking about ancestral or derived condition for the Oceras, and also about termites. So I'll just go on to my second question. And I think what you were just talking about relates to this in terms of the different origins of sociality. But for example, in bumblebees and in other eusocial species, we know that foundresses, in some other, not all obviously, that foundresses do brood care up until the time that the first workers emerge. And I'm thinking of bumblebees just as one example. And I'm wondering if anything along the lines of what you have done has been done with any of those species, because they would seem to be, in a way, simpler models with a different evolutionary origin. But it would be interesting to see if the phenomena that you're observing in Lasius in terms of brood care and presence of workers and the flexibility. And of course, in the workers, we know that in terms of age polyphonism, they can go back and forth in terms of the work that they do, depending on the social context. And so I just wondered about that. But basically, I thought your talk was absolutely fascinating. I loved it. So thank you very much.
Yeah, thanks. It's a good idea. I mean, we do have a new PI here in the department, Joe Colgan, who is working on bumblebees, and that's something that we've already discussed a bit. That it would be fairly easy to do so. So basically, to also disentangle the seasonal effect from the social effect, because in that case, they always get workers at the same moment in their yearly life, and we could make sure that they don't get workers, or that they get workers before they lose the workers, and see the effect on the bumblebee queen. That's a very good point. I think that's something that would be super interesting to look at. Yes.
Thanks.
Then there is another question from YouTube. Fran Lapron is saying, Thanks for this talk, Romain. Maybe it's not a matter, but about from 25 of your queens, 3 died. The question is do you think there's the same mortality in the field like in the lab? And is 12% mortality, do you think this is high or low?
It's very low because it's in the lab. So probably the success rate of finding a colony in the field is, I mean, I don't want to give a specific number, but it's super, super small. Probably maybe 1-2% of the queens that fly out actually manage to successfully find a colony. But that's mostly because of extrinsic mortality, because of birds during the nuptial flight, because of rodents on the ground, because of scientists collecting them for experiments, or people working, stepping on them, and also because of pathogens. And all of these are very limited when you put them in a tube in the lab. And the mortality we see, usually they have a lot of fungus growing on them, either after they die or because of that they die. It's difficult to tell. So that could be linked to simply queens being affected by a pathogen. But basically in the lab, if they survive, so as I said, we collect hundreds and hundreds of queens every year, and we have more than 90% of them that basically manage to produce workers. And the 10% that don't manage to do so, I would say that 3/4 of those die in the first week. So they basically die right away, or they cannot produce, and the rest cannot produce eggs. And we have some cases of queens that produce always eggs, but they never develop further. And that could be maybe because they are haploid eggs and they're not supposed to, and they are not mated, and therefore, well, if they lay haploid eggs, they could still develop to larvae. But we know they also produce trophic eggs, so eggs that have no embryo in there. So maybe that's what those queens are producing. And I'm not sure, but we do have dysfunctional queens that do not die at the beginning and do not manage to produce workers. And if usually they don't manage to even produce larvae. But this is a very, very small proportion of the queens. Most of them either manage to do it or they die right away in 8% of the cases, let's say.
If I can follow up on this, is this the case for most of the ant species you have in the colonies, or is it specific for the brown garden ant, what you just described?
That's the only species I work with that I collect founding queens. The other species I work with, we either collect established colonies or they're clonal, and we can just keep them in the lab for a long time. But generally speaking, I think the low success of colony funding in the field is something that is pretty well documented, that basically colonies have to produce thousands of new queens to only get a few that manage to produce a new colony in the end. But in the lab, I guess the mortality is always lower than in the field, but I don't know if it's to the extent of what we see with the lasers.
Yes, so thank you again very much for a fascinating talk and a very interesting discussion. It was very interesting for me. It was a great ending of this autumn seminar series, and as there are no more questions, I'm going to terminate the live screen on YouTube now. And but still, thank you very much from everyone here and for everyone.
Thanks for the invitation and for organizing everything. That was a great series, and I hope it will be the same for the next session.